do treatment of hypoxia. So if you have any questions in between, so don't hesitate, just ask. That we have some interactive discussion going on. So based on the journal clubs last week, well, at least in the journal clubs I did, we talked about the hallmarks of cancer. And then I was asking to some of you people, how many hallmarks are there? Everybody convinced, yeah, there are 6 hallmarks of cancer. So no, there are not 6 hallmarks of cancer, there are now 10 hallmarks of cancer. So I updated Jan's slide, which still had the 6 hallmarks of cancer. And I'm showing you now the 10 hallmarks of cancer. So it's by the same authors, Hanahan and Weinberg, and it has been published in 2010. So it's 10 years after the original 6 hallmarks of cancer. So the original 6 hallmarks of cancer are depicted over here. So you have unlimited proliferation, you can evade growth suppression, so that means that you have an unlimited amount of growth factors present. Invasion metastasis is activated. You can enable the replicative immortality, so cells keep on growing. Your cells become resistant to cell death, so anti-apoptotic pathways are enhanced. And then we have the big question mark over here, which I'm going to discuss later on. New hallmarks of cancer are deregulated cellular energetics, so glycolysis, the main factor of getting ATP, of getting energy, is by using glycolysis. There's also a new hallmark of cancer, avoiding the immune destruction. And closely linked to that is tumor-promoting inflammation. And then the last new one is then more a genetic, epigenetic point of view for the hallmarks of cancer, and more in particular the genome instability and mutations. All the other ones around are possible treatments that are currently in clinical practice to address several of these hallmarks of cancer. But well, these are not part of the lecture of today. So what I want to go into detail today is about the last hallmark, or one of the initial 6 hallmarks of cancer. It's inducing angiogenesis or neoangiogenesis. And this is closely in relationship to hypoxia, which will become clear throughout the lecture. And this induction of angiogenesis can be blocked using VEGF inhibition. So I'm going to show you also some slides on that. So now, what's hypoxia? At the left side, there's I depicted here a sort of cartoon about the vasculature in normal healthy tissue, like a liver or a kidney or a lung. So you can clearly see that these vessels are mature, well structured in lines, some branches, but all of them are sort of straight dividing network. If we're comparing that to a tumor on the right side, you immediately see that these vessels, well, don't have a straight structure. There are bends. You have spots in the vessels where your red blood cells accumulate because the flow is reduced over there. You have some blind ends, so a vessel is started but just doesn't grow further on. There's also a possibility to have a temporary occlusion, so that your vessel shuts down. So all these factors cause that the oxygen delivery, which in essence comes from the lung into the vessel system and goes out the vessels into the cells, is sort of hampered comparing with this situation. So we notice in a lot of cancers and a lot of tumors that at several layers of your vessel, a functional vessel, at several layers of these vessels, that oxygen gets deprived. So you get the formation of hypoxia or low oxygen regions. And why is that? That's because of the diffusion distance of oxygen, which is like around 150 micron. So all cells beyond the 150 micron, in a sense, don't see any oxygen, so it becomes hypoxia. Some more characteristics about hypoxia is that in a large percentage of human solid tumors, we find these hypoxic regions. And we noticed during the last 20 years, so to speaking, that most of these hypoxic cells are resistant to standard therapies. And I mean with standard therapies, surgery, chemo, and radiation therapy. On the other hand, since these hypoxic cells are only found in tumors or in cancer and not in normal cells, it's a way to exploit this. So we can address treatments specifically towards these hypoxic regions in an attempt to cure cancer cells and tumor cells. And well, probably it's one of the best validated targets within oncology so far. There are, in essential, if we have to separate hypoxia in two classes, you have two separate ways to look to that. So the first way I sort of described already. So you have a functional blood vessel, and these are the tumor cells around that vessel. So how far do you go away from the vessel, how lower the diffusion is from oxygen into the tissue, so like depicted over here with these arrows getting smaller and smaller. So at a certain point, cells don't see enough oxygen anymore and become hypoxic. So we call the diffusion-limited hypoxia or chronic hypoxia. This is with a functional blood vessel. So on the other hand, you can also have a blood vessel which is temporarily occluded, so it's a partial shutdown of the vessel. So that means that even the cells in the close vicinity of these vessels don't see oxygen anymore because the blood vessel is closed, so blood cannot pass anymore. Red blood cells cannot pass anymore, so there's no oxygen delivery anymore. So we call this perfusion-limited hypoxia, since it's due to a hampering in perfusion, or another term is acute hypoxia. So how do we have to see this hypoxia existence within the phenomenon of cancer initiation and progression? So if you're looking to the top part of this figure, so this is a normal cell, you have a blood vessel, you have a lymphatic system. So in essential, this is how most of your organs, well, I hope like to say all of your organs look like. So for the smokers over here, or for people who like to sit in the sun a lot in summer, or go in winter to, I don't know, some fancy warm country, you can get UV damage as well. So sun can cause UV damage. Cigarette smoke, so for all the smokers, don't do that anymore, please. You can get a rise of mutations in these normal cells. So this normal cell will develop into an abnormal cell. So, and this occurs for years and years and years and years, until a certain moment that you have so much abnormal cells that they form a sort of abnormal amount of cells, like a small neoplasm. So we call that a cancer in situ. So this is depicted over here. So you have an abnormal clump of cells, so to speak. And at a certain point, these cells will follow the hallmarks of cancer and will lose the limit to not proliferate. So they will start proliferating all the time, so without any restrictions. So how do we get now from here to an invasive or a malignant cancer, so which is depicted more in detail over here? We see over here, so the blood vessel, and this tumor attracts these blood vessels. Blood vessels go into the tumor. The moment that you have these blood vessels inside the tumor, there's an escape route for these cells. So these cells can break, your cancer cells can break through the blood vessel and travel through the blood vessel and the lymphatic system to form metastasis. So this is a characteristic of a malignant or an invasive cancer. So what's triggering this? In essence, the trigger for that is the angiogenic or the hypoxic switch. So we'll go more into detail later on that. So this is a sort of other depiction on carcinogenesis. So they have a normal epithelial boundary, and this develops into an interstitial neoplasia by smoking or by UV. For example, there are other examples as well. This develops to cancer in situ, so the clump of cells, which can become invasive and give rise to metastasis. So the physiological state over here, we see, so as second, that intermittent hypoxia and HIF, so hypoxia-inducible factor stabilization, plays a role in this. And also those two lead in essentially to glycolytic phenotype, so one of the new hallmarks of cancer. So looking more into detail on how this occurs, I don't know, I'm first going to put on something else. So to see it, if you get slides from somebody else, it's sometimes difficult. Anyway, so we know that there are hypoxic cells or cells with low oxygen in the tumors because, well, if you have a clump of cells, at a certain moment, also this passes beyond the diffusion distance of oxygen. So several years ago, people investigated this by putting electrodes, oxygen electrodes, inside the tumor and inside normal, healthy, matched tissue. So if you have a breast tumor, they insert these electrodes in the tumor of the breast, and they insert also these electrodes in the normal, healthy breast. So what they noticed, and this is sort of like over here, so comparing, so in normal tissue, they noticed that there is a quite high oxygen concentration. Then this is expressed now in millimeter mercury. And when they compare that with oxygenation in the tumor, they notice that indeed the oxygen concentration in the tumor is much lower. If we're looking more into detail to those figures, so this is from a normal, healthy cervix. So you see that oxygen concentration can vary in essential from 0 to 100 millimeter mercury. So you get a big variation with a sort of peak over here at a similar range like over here for the breast. Comparing with cervix tumors, in essential quite similar, is that you get your peak shifts to the left, to the lower millimeter mercury range. So your oxygen concentration levels in cancers are much lower compared to normal tissue. Again, supporting the idea that if you have too much cells as a clump together, that you get beyond the diffusion distance of oxygen. So there have been over the past 10 to 20 years a lot of these studies in several tumor types. So I'm not going into detail on all those, but we noticed that, so if you, from the previous slide, so let's say the average oxygen concentration in healthy tissue is 40-50 millimeters of mercury. So if you keep that in mind, we notice that for all these several types of cancer, that oxygen concentrations are much lower, are really significantly lower. So this does not only count or occur in cervix or breast cancer, but also in brain cancer, head and neck, lung, pancreatic, prostate, or tissue carcinoma. So it's the characteristic of having low oxygen levels in the tumor is a broadly spread phenomenon through solid tumors. So how does it look? So this is an example of a staining of a tumor. What we see over here in red are the blood vessels, and in green are the hypoxic areas. So to measure hypoxia in staining, we use pimonidazole. So this is a drug which is specifically trapped in hypoxic cells. And then with an antibody, we can detect this marker. So what we see over here is that although we know there's a lower oxygen concentration in the tumor, we notice that there's a heterogeneity in this oxygenation in tumors. And the heterogeneity is seen in the amount of hypoxia or low oxygen concentration regions, like we see over here. So in this tumor, there's only a small amount of cells who are hypoxic, while comparing with this tumor, the different tumor, you have much more areas of hypoxia. So there's a heterogeneity in amount. There's also heterogeneity in severity. So the cartoon over here is showing a vessel with tumor cells around it. So this is again the principle of your oxygen diffusion. So how farther away cells are located from the functional vessel, how lower the oxygen concentration will be. So there's also heterogeneity and severity of oxygenation, of hypoxia. There's also heterogeneity in distribution. So this is a section of the tumor, and we notice that like in the middle of this section, or inside the tumor, there's an area with a low amount of green, so a low amount of pimonidazole or hypoxia. While, for example, the left side and the right side show much more hypoxic areas. So there's also heterogeneity in distribution of your hypoxic areas throughout the tumor. And as last, there's also heterogeneity in timing. So what has been done over here is we injected 2 hypoxia markers, but at a different time point. So the first one is depicted over here in green. This is pimonidazole. So we injected that in a patient. We waited for 24 hours and then injected a red lower hypoxia marker in red then to see the dynamics of hypoxia. And what we noticed in this tumor is that this hypoxic fraction seems to be quite stable in this tumor. So we see green areas, which indicates that at the end, after 24 hours, there was no red staining, so these cells are not hypoxic anymore after 24 hours. The red, this is just single red, indicates that cells at the injection of the first marker were not hypoxic. These were only hypoxic at the moment that you injected the red marker. And then the dual, so the combination yellow, is that these cells stay hypoxic during this whole 24 hours. And in this case, we see that the majority stains yellow, so this is a quite stable hypoxic tumor. So we have heterogeneity at 4 different levels: in amount, severity, distribution, and timing. So how does that look in patients now? So we selected 6 different patients, all having had a neck tumor, and we did in essentially the same analysis as shown on the previous slide. And we noticed that, well, this patient has sort of no hypoxia, so the hypoxic fraction was only 0.3%. This patient had 5%, 7%, and you see that some patients have even up to 17 to 20%. And not only shows this that within the same cancer type you have heterogeneity in amount, so going from 0 to 17, you see that also the distribution is different. So this patient had a more focal, sort of bowl-shaped hypoxic areas, while this patient had really strengths of hypoxic areas. So within one patient population, what we see as heterogeneity in oxygenation, this is true for any patient. Why is hypoxia now important, clinically seen? So what are the consequences of hypoxia? So there has been published a study in 2005 with almost 400 head and neck patients collected from different institutes, where they, using these electrode measurements, they measured the amount of hypoxia in the tumor. And then they split these patients having a low amount of hypoxia in the tumor compared to a high amount of hypoxia in the tumor. And noticed, and when looking to survival, so this is a follow-up of 3 years, they noticed that there's a clear split in survival. So survival is depicted here on the y-axis, and showing that the more hypoxic tumors, so patients with more hypoxic tumors do significantly worse in outcome, so they die faster compared with patients who have tumors which are less hypoxic. So hypoxia is really clinically important, and that has been shown in not only this big study with a lot of centers, but also for head and neck and several different institutes, for cervix in different institutes, and also for a lot of other tumor sites. So looking a little bit more into details on the head and neck tumors, we did an analysis of these almost 400 patients and looked to the hypoxic fraction. And we noticed that by this Eppendorf electrode, so the oxygen electrode measurements, that we saw a variation or a range of being non-hypoxic, so 0% of hypoxic, up to 100% hypoxic. So the hypoxia itself varies a lot amongst the different patients. So why is it now clinically important? Well, it causes resistance to the standard care. So it causes resistance to chemotherapy, it causes resistance to radiotherapy, and it contributes to malignancy as shown before, and as I will show you later on as well. So how does it cause resistance to chemotherapy? So I took back that slide that you saw, or that cartoon that you saw before, functional vessel, tumor cells around it, so how further on, how lower the oxygen concentration gets into the tumor. This also means that how further on your cells are located from a functional vessel, how lower the amount of drug concentration will be. And chemotherapy is a drug which is injected intravenously through the vasculature. So how farther away your tumor cells are located from a vessel, how lower the amount of drug they will see. So how lower the amount of cure will be. So that has been depicted over here. So if we look to the distance from a capillary, so how further on away, and to comparing that to the surviving fraction, essentially this is wrong. It should be the other way around. So how further on of a vessel, how lower the hypoxic fraction should be, how lower the survival should be. sorry. No, I'm wrong. See, yeah, that's when having not enough time to prepare. So how further away from a capillary vessel, how lower the amount of chemotherapeutics will be for these cells. So how more the cells will survive. So how less efficient your therapy will be. So the cell killing by these chemotherapeutics or anti-cancer drugs will be reduced as a function of distance from the capillary. I hope that's clear. Yeah. I messed it up sort of myself. So what's now important in this resistance to chemotherapy? Well, one, the microenvironment, so everything surrounding the tumor. And then more particularly the reduced perfusion. So how lower the perfusion is, how lower the delivery will be of your chemotherapeutic drug. It's depending on the microcirculation or circulation in general. And it's also dependent on reduced pH. This reduced pH, I will come back to that in detail later on. It's also dependent on your tumor cells. So tumor cells located further away from a vessel are growing slower because oxygen concentration is lower, the nutrients they get out of the vessels is lower, so they grow lower, they grow slower. And this is now one of the characteristics of chemotherapy. Chemotherapy attacks highly proliferating cells as well. So if you have reduced proliferation, your chemotherapeutics won't be as effective as for cells which have a normal or high proliferation. And also tumor cells can activate a lot of additional systems to induce resistance to these chemotherapeutics. So going a little bit more into detail about this cellular proliferation. So this is also again a section staining with the head and neck cancer. So what we see over here is in red the blood vessels. So everything gets delivered from out of the blood vessels: oxygen, nutrients, chemotherapeutics. If we look a little bit further away from these vessels, we see proliferating cells, so depicted in purple. If we go beyond that, so still even more away from a vessel, hypoxic cells arise. If we're going too far away from a blood vessel, you get cell death or necrosis. So we notice indeed that in the areas of low oxygen or highly hypoxic cells, proliferation is much lower compared to areas which have a normal amount of oxygen. These cells grow much faster or have a normal proliferation. So there's an inverse relationship between hypoxia and cellular proliferation. If we're looking to radiotherapy, so again the same cartoon, so cells further away from blood vessels see less oxygen. This is the principle of radiotherapy. So you will see that in the next slides. So again, we see that how farther away from your capillary, so how farther away from the blood vessel, cells see much lower oxygen concentration. Radiotherapy is less effective, so cells are surviving much better. So also for cell killing by irradiation, this depends or is a function of the distance from the blood vessel. So how does radiotherapy work? And you will get an extended lecture in the next block on radiotherapy, so I will keep this short. If you irradiate, an electron jumps away from the atoms, and this can interact immediately, so a direct interaction with your DNA and causes DNA damage. But there's also an indirect action. So this electron can hit a water molecule, as seen over here, causing a radical, and this radical interacts with the DNA. So to fix the DNA damage caused by these radicals, you need oxygen. So you need normal oxygen levels to fix these levels. And this is depicted with the reaction over here. So you get your radical, causes DNA damage. Together with oxygen, it forms a stable end product, which then can be cured by DNA repair pathways. So within radiotherapy, oxygen stabilizes your lesions at the DNA. So that's why oxygenated tumors, so tumors with a lot of oxygen or no hypoxia, are three times more sensitive to radiation. So you will see that in the next slide. An additional feature of tumors is that they DNA repair mechanisms show mutation in that. So they are not capable anymore of repairing the DNA damage caused by radiotherapy. So in that sense, combination of having oxygen and mutations in your DNA repair pathways determines the success of radiotherapy. So how do we have to see that in conjunction with oxygen and radiotherapy? So we plotted here the surviving fraction in comparison with the dose of radiotherapy. So this is shown in gray. And we did 5 conditions. So we exposed cells to 5% oxygen and went lower. So from 5 to, in essential, 0.01% of oxygen, or almost no oxygen anymore. So we started at 5%. Why? The air that we're breathing at the moment has 21% of oxygen. However, so that's what we have in our mouth and in the bronchi and in the top of the lungs. So how farther you go inside the body, this oxygen level will reduce from 21% to approximately 5%. So if you check oxygen concentration in the liver, for example, you will notice that this is only 5%. So that's the reason why we started with 5% and not 21%. So if we then look to the survival curve, you see that there's a shift to the right with decreasing oxygen concentration. So that means that, like for example, if we want to have 99% of our cells killed, or even more, 99.9% of our cells killed, you see that if we have lower oxygen concentration, that the dose to get the same level of cell kill needs to be much higher. And if you calculate that, and that's what you can see over here on the next slide, so this is in oxytic or 5% oxygen concentration, comparing hypoxia or anoxia, so no oxygen concentration left. You see that this, the difference between the effect from going from hypoxia to oxic concentration, and compare that to the effect of no irradiation to a certain dose of radiation. So we call that oxygen enhancement ratio. So this is the orange divided by the blue. This is 2.8. So there's a difference of 2.8-fold in resistance of irradiating cells upon hypoxia comparing with regular oxygen concentration regarding survival. So to say it in another way, so to kill the same amount of cells in a tumor, if this tumor is hypoxic, you have to give 2.8 or almost 3 times a higher dose compared with regular oxygen levels in the tumor. So, regarding clinical outcome and tumor hypoxia, I showed already some data on that these hypoxic tumors can lead to more aggressive metastatic tumors, and there's a reduced effect of 3. So both on chemotherapy, which involves reduced proliferation, reduced delivery, also acidosis and multi-drug resistance. And for radiation, it's a reduced oxygen enhancement and then a reduced apoptotic potential, which I will show later on. So, next part is what determines tumor hypoxia in a cell. So in essential, you can summarize that as selecting cells which are hypoxia tolerant, so which are able to survive upon hypoxic conditions. And a second quote, or what determines tumor hypoxia, is the initiation of adaptive responses downstream. So we will go a little bit more into detail about the molecular responses upon hypoxia. So how does that in general? So to select hypoxic tolerant cells. So this is the vessel, you have tumor cells, so further away from the vessel, these cells become hypoxic, and in essential, these cells will die through apoptosis. This is p53 dependent, the apoptotic cell death, further away, so in hypoxic cells. Now, what occurs in most of the cells is that p53 is mutated. So if we look at a patient tumor, we see that 95% of the tumors are p53 mutated. So these cells cannot clear anymore or cannot die through apoptosis, through p53 dependent apoptosis. So this is how it starts. You get sort of one cell which shows a mutation in p53. So when this tumor is growing, you will see that hypoxic cells with a wild type p53, well, they will die from apoptosis. the other cells with a mutation in p53, well, they cannot die, so they keep on surviving, they keep on growing. And at a certain moment, they will repopulate the whole tumor, like you see over here, with only p53 mutated cells, independent of being hypoxic or not. But since this is a phenomenon important in hypoxia-induced apoptosis, we end up with a tumor which is tolerant to hypoxia. Because if you now expose these tumor or having hypoxic areas in this tumor, they won't die anymore due to p53 dependent apoptosis because your p53 is mutated throughout the whole tumor. So this is one way, one possible way to raise a hypoxia tolerant tumor. If we look more into detail about adaptive responses, this is sort of the general summary of all adaptive responses upon hypoxia. So I'm not going to go into all details of those, so I don't expect that you know all individual gene and pathway on that, as long as you know the general idea about it. So in essence, there are three adaptive responses. One is regulated by HIF1 alpha, so hypoxia-inducible factor 1 alpha. The second one is, so in that sense, hypoxia inhibits the degradation of HIF1 alpha, so I'm going into more details later on. The second part of an adaptive response is the upregulation or the stabilization of the UPR, so the unfolded protein response, who will have an effect on transcription. So comparing that, HIF1 alpha has only an effect on transcription. UPR has an effect both on transcription as on translation. So both on conversion from DNA to RNA and the conversion from RNA to proteins. A third adaptive response is a translation response, and this is what you all saw last week in the journal club, is the mTOR pathway, which can be blocked upon hypoxia. So all these three pathways, independently or working together, lead to hypoxia tolerance. And this can influence then metabolism, angiogenesis, autophagy, another way to get rid of, or another way to remove dead cells, and endoplasmatic reticulum homeostasis. So, if we go more into detail about the mTOR pathway, and essentially I shouldn't do that anymore because you guys all know it since last Friday. So this is the extended version of the mTOR pathway. So you get PI3 kinase, who influences AKT, which activates the TSC1-2 complex, all going down to mTOR. You get activation of S6, phospho-S6, you get phosphorylation of 4-BP1, so you guys all know that. So how does hypoxia come into the picture on this? In essence, hypoxia blocks mTOR, activates AMPK, activates the pathway from 4E to the conjunction to 4ET. So not the promoting what actually should be is that 4E starts translation, so cap-dependent translation. So hypoxia shifts 4E, even if it's free. If 4EBP1 is phosphorylated, 4E gets free. So even if it's free, hypoxia diverts 4E to form a complex with 4ET, and translation stops. So hypoxia suppresses mTOR and all downstream effects. So metabolism, growth, proliferation, survival. If we look into the UPR, unfolded protein response, so hypoxia affects all three arms of UPR. So this is the PERK arm, the IRE1 arm, and the ATF6 arm. So I'm not going into detail on that. I'm going to show later on some examples of the specific pathways, how you can target these different pathways. So if we're looking into the PERK pathway, one of the components is EAF2 alpha. When this one is phosphorylated, it can translate mRNA. So what it shuts down mRNA. So what we did is we made a kinase dead mutant, which phosphorylates but cannot do anything anymore. So we notice that upon anoxic exposure, that this phosphorylation is enhanced. When we look to the IRE1 pathway, the final product is XBP1, which needs to be cleaved to a soluble form. So if you expose cells to anoxia, several hours, we see that there's an enhancement in this soluble XBP1. So activation UPR through IRE1 to get soluble XBP1. And as last as an example, if we're looking to ATF, if you so this is the lower plot, this is the actin, the loading control. So if you look to the ATF, several hours of hypoxia, ATF gets upregulated. So hypoxia activates all three different arms of UPR. Going a little more into detail about the HIF activation in tumors. So what happens upon normoxic exposure? So HIF1 alpha has proline residues in his C-terminal end. So upon normoxic conditions, you have the prolyl hydroxylases, which can hydroxylate the proline on HIF1 alpha. So you get HIF1 alpha not with proline but with hydroxyproline, so this OH. So when your HIF1 alpha shows this OH, it forms a target or substrate better for the VHL, so the Van Hippel-Lindau protein. So the Van Hippel-Lindau protein recognizes this OH, forms a complex with HIF1 alpha. The moment that this complex is formed, you get the complex gets pushed to the ubiquitin proteasomal degradation pathway. So upon normoxic conditions, PhDs hydroxylate HIF1 alpha, VHL grabs this OH, and HIF1 alpha gets degraded. There's also another possible way, not through proline, but through arginine, also to the C-terminus, in which FIG, so this is the factor inhibiting HIF1 alpha, also does the same thing towards proteasomal degradation. So what do we see now upon hypoxic conditions? These HIF hydroxylases are inactive. The same with the FIG, so the factor inhibiting HIF is also inactive. So in essence, HIF cannot get hydroxylated anymore. So it doesn't form a target anymore for VHL. You get HIF1 alpha gets not degraded through the ubiquitin proteasomal pathway, but it's free, travels to the nucleus, forms a complex with HIF1 beta, and will sit on the HRE, so the hypoxia responsive elements in the promoter regions of several genes. So I'm going back to that. Except from being normoxic or hypoxic in the tumors, there's a third way to activate HIF. This is the oncogenic pathway. So in this oncogenic pathway, PhDs are active, FIG is active, so you get hydroxylation of HIF1 alpha. So that means that HIF1 alpha remains a target for VHL. So the oncogenic activation of HIF occurs through the VHL. This VHL gets mutated, so it looks different compared with those two VHLs. It's mutated in such a way that it cannot recognize HIF anymore. So HIF stays in a hydroxylated state, but gets not recognized by VHL for proteasomal degradation, and can also travel to the nucleus to start transcription. So if we're looking in a panel of breast cancers, we did, other scientists did HIF immunostaining, so this is an antibody directed against HIF. And we see here from a healthy breast going from B, C, D, E, F towards an invasive carcinoma. And what we see is HIF, when it's active, it goes to the nucleus, so you expect a nuclear staining. So that's what we see starting from the DCIS. For sure in the invasive carcinoma, all these brown dots in the nucleus. So a lot of these breast cancers express HIF1 alpha. So if you split the patients having HIF1 alpha, more than 5% HIF1 alpha staining, compared to the patients with below 5% of HIF1 alpha staining, you can see that there's a significant split in survival rates between those patients. So patients with a lot of HIF1 alpha do significantly worse, so they die sooner, and more patients die at the end. So this is not only true for the breast cancer, but it's true for a whole range of cancers. And you see that in most of these cancers that HIF1 alpha gets, when you have increased HIF1 alpha, that it leads to an increased mortality. And there are a few exceptions, like there are reports for lung non-small cell lung cancer that you get a decreased mortality. So that means that it would be good to have HIF1 alpha overexpression. And another one is head and neck squamous cell carcinoma, after surgery. That is also a decreased mortality. But in general, if you have high HIF-1 expression levels, the mortality is increased. So it's not good for patients. So if we're going more into detail on this HIF activation in tumors, so HIF can be activated upon hypoxia, but also upon oncogene activation, or the other way around, tumor suppressor inactivation, HIF-1 gets stabilized. So at the end, what does HIF-1 do? It leads to tumor growth, invasion, and metastasis. So now we're going to go into detail on how this occurs. One of the possible ways is through stimulating angiogenesis. So we're going to look now a little bit more in detail about VEGF. So VEGF is a pro-angiogenic factor. So it induces angiogenesis. And VEGF gets upregulated, or the expression of VEGF gets upregulated if you have cancer cells, so this is this line. If you have tumor-promoting inflammation cells, so this line. Also from the bone marrow, so this part. So they all lead to the formation of endothelial cells. So going even more into detail, so this is the pathway of VEGF. So VEGF as a ligand binds the VEGF receptor and then activates a whole bunch of pathways to induce angiogenesis. One of those pathways is again AKT. As I showed to part of the group, the scheme with AKT, that AKT does not only signals to mTOR but also to apoptosis. AKT also signals to VEGF or angiogenesis. So if you have VEGF overexpression induced by HIF, you get an endothelial migration, proliferation, and survival. Your vessels get dilated, they are more permeable, and this all leads to an increased metastatic potential. One of the treatments is bevacizumab or Avastin. So this is an antibody, a monoclonal antibody, which in essence blocks the receptor, so the VEGF receptor, in such a way that VEGF itself cannot bind anymore to its receptor. So these are some data from breast cancer. In essential, what we see is that patients with metastasis show much more or higher microvessel count. So the amount of angiogenesis is much higher in patients with metastasis. If you compare it with normal tissue, it's only VEGF that shows an increased expression. So this is tumor. The white ones are normal tissue. Like for other ones, like TGF alpha, TGF beta, FGF, there's no difference. And if you split the patients, again, having high versus low VEGF, so the patients with high VEGF do significantly worse. So this is the blue curve. So they are associated with increased metastasis and with poor survival in breast cancer. Not only in breast cancer, but in much more. Another way from HIF, starting from HIF, to induce tumor growth, invasion, and metastasis is a metabolic adaptation. Here I'm going to show you the example of CA9, so carbonic anhydrase 9. So it has been shown that hypoxic tumors produce acid. So the extracellular environment of hypoxic tumors is acid. So that means having a pH lower than 7. How does that happen? So upon hypoxia, cells shift their metabolism from the TCA cycle, so from a metabolic respiration towards glycolysis. Result of that is that there are only 2 ATP molecules available instead of the 38 that you would expect in normal healthy cells. But one characteristic of a tumor cell is that he will do everything trying to survive. So he has lower energy availability. So what they will do is they will increase the glucose uptake, and this is done through a HIF-dependent manner. So your glucose transporters will increase. You get more influx of glucose in your cells, in essentially to raise the glycolysis. So to have more ATP in that way to trying to survive. Problem is that you only have glycolysis. So the end product of glycolysis is pyruvate. Pyruvate usually goes to the Krebs or the TCA cycle and gets converted through several steps, delivering ATP. So what happens if you have only glycolysis is pyruvate shifts to lactate. Lactate is in essentially an acid. Tumor cells don't like intracellular acids, so they will upregulate a whole bunch of systems to get rid of this acid, all measurements trying to survive. Now, one of those are, this is NHE1, so this is a sodium proton co-transporter, MCT14, which transports lactate out of the intracellular environment, and C9, so carbonic anhydrase 9. So carbonic anhydrase 9 takes the acid, so CO2. CO2 diffuses outside the cell, interacts with water, and through the activity of Ca9, it gets converted in bicarbonate and proton. Proton stays outside the cells. Bicarbonate gets exchanged with chlorine and goes inside the cell again to again capture one of these protons to convert again to water and CO2. So it's really a cycle. So what have people done is they stained sections. So this is again a breast tumor for HIF1-alpha, as we showed before. And again, also they stained for C9 and GLUT. So both C9 and GLUT over here, C9 over here, are HIF targets. And they also saw in sort of the same regions as where HIF1-alpha is expressed, C9 expression or GLUT1 expression. So the C9, this is a member of the Ca family, so the carbonic anhydrase family, and in essentially there are 15 members. So I'm not going into detail about all these members. What you need to know about C9 is this a transmembrane protein, so it has a short intracellular part and a large extracellular part. And so the bulk here over here is depicted over here. This is the C9 active site. So there in that part, this reaction occurs. So C9 has been shown to be expressed in broad range of tumors. So this is, for example, and this is a vessel. The blue is just counterstaining with hematoxylin, as seen in the paper from last Friday as well. If you go away from this blood vessel, hypoxia arises, or C9. So hypoxia arises, HIF1-alpha gets stabilized, C9 expression gets upregulated. So that's what you can see here in this brownish staining. And this is necrosis, so this is cell death, so beyond viability. But what's more particular about C9 is if you compare over here on the right side, these are all carcinomas. And on the left side, these are the healthy organs from which these carcinomas arise. So this, for example, lung carcinoma, well, occurs in the respiratory tract, obviously. So but if we check these normal tissues for C9 expression, well, virtually none, except for stomach and duodenum, don't express C9. Compare that with the carcinomas, the tumors, all of them, or almost all of them, express C9. So C9 is a marker really specific for tumors. So, and if you split patients again, having low versus high C9 expression, or having low versus high extracellular acid, so this is low extracellular acid, this is a high extracellular acid, you clearly see that we can split patients into two groups, a good surviving group and a bad surviving group. So the last thing I want to talk about for the HIF pathway is actually which proteins or which genes are involved in this invasion and metastasis. And one of those is so the mechanism. So I told you already that if you have hypoxic cells, genes get upregulated, VEGF gets upregulated, you get neoangiogenesis. Having these new blood vessels, they are abnormal in structure. So cells have the ability to go through these abnormal structures and to escape their initial primary tumor and to travel around to the system, to the vascular system, until they are stuck somewhere to form a metastasis. So how does that work molecularly? Like this. So you have hypoxia, upregulates HIF1-alpha. So HIF1-alpha can upregulate VEGF, C9, that's what I showed you in the previous slide. Can also upregulate EMT regulators like Z1 and Z2. It upregulates twist, LOX cell 2, several more. And these, mainly EMT regulators and twist, are involved in EMT, so epithelial to mesenchymal transition, which leads to invasion, intravasation, and if you take that on, it leads in the sense to metastasis. While LOX, for example, when that is upregulated, there's an unknown factor in between that we don't know yet, but it's sort of more direct link to metastasis. So if we're looking a little bit more into detail about this twist and EMT, so this is a normal epithelium, so we have cobblestone cells within polar and the basal cell line. Nucleus is in the middle of these cells. So when EMT occurs, this cobblestone cells shift to more like stretched fibroblast-like cells. And we know that Twist is involved in this. So if you have this EMT, again, further induced by Twist, cells can get loose or much looser conformation compared to these cobblestones. So they can intravasate blood vessel, travel through the blood vessel, extravasate again for micrometastasis. And the moment that these micrometastases undergo MET, so that's the opposite as EMT, so mesenchymal epithelial transmission, they form this micrometastasis, and this will at the end up again with sort of cobblestone-like cells. So this is upregulated upon hypoxia, this whole process, and one of the most important proteins which are affected by these twists is ecadherin. And ecadherin is involved in the tight junctions of these cobblestone cells, so they're sitting in between the different cells. So what happens is that the twist blocks ecadherin. So you don't have these tight junctions anymore. So we did that. That's very clear, but at least on my screen it's very clear. So we compared normoxic with hypoxic exposed cells. So you have to believe me if you don't see it. We did a HIF1 alpha immunostaining and ecadherin immunostaining. So we noticed upon normoxic exposure, HIF1 alpha is expressed to a very low extent. Ecadherin is expressed to a very high extent. So it forms really the tight junctions between all these cells. If you expose cells to hypoxia, HIF1 alpha gets upregulated to the nucleus, and ecadherin expressed is just gone. It's lost. So you don't have these tight junctions anymore, and cells can undergo EMT. So I showed you examples of 3 genes, or 3 proteins. So VEGF, C9, twist with ecadherin, so the more metastasis. That's not the only thing that hypoxia does. So the hypoxia does not only influence those 3 genes. What we did is we did a microarray study to figure out all the genes which are influenced by hypoxia. And it would take me several weeks to discuss those all. So what we did is we made a sort of summary in 3 different cell lines. We noticed that a lot of cells, a lot of genes, so several hundreds of genes are upregulated. And on the other hand, several hundreds of genes are also downregulated by hypoxia. So you have a bivalent system, upregulation of some genes and downregulation of other genes. So if you group these genes, so to make sort of hypoxia signature, so only the genes which are importantly influenced by hypoxia, and you give a score to this grouping, a low score, so low influence, or a high score, high influence, we saw in a breast cancer meta dataset, so this is a dataset with more than 2,300 patients, that if you have a low score, so the blue one, comparing with the high score, so patients again with a high score have a significantly worse outcome comparing with patients with a low score. So this is just as sort of pointing out that not only those 3 genes that I discussed, but hundreds and hundreds more of these genes are influenced. So as a last part of the lecture, I want to discuss some strategies to overcome tumor hypoxia. So how can we treat tumor hypoxia? So you saw this cartoon already. So how are we going to treat? Well, one obvious way is we're going to replace oxygen in the tumor or restore oxygen in the tumor. So how can we do that? With EPO, for example. So EPO has been used with by Lance Armstrong for other reasons than restoring the oxygen balance in the tumors. ARCON, that stands for accelerated radiotherapy, in combination with carbogen. Carbogen is 95% oxygen, and nicotinamide. Nicotinamide is a vasodilatator. And another example is an oxygen mimicker, nimorozole. Another way is exploit tumor hypoxia. So by using bioreductive drugs, which are only trapped in hypoxic cells. An example of that is pimenidazole, but pimenidazole has no toxic effect. So if you have a drug which is able to get bioreduced, it sticks in hypoxic cells to execute its toxic effect. Or anaerobic bacteria. So I will go into detail on the anaerobic bacteria. And the third way is to target the biological responses. So I showed you UPR, the HIF, and the mTOR pathway. So these are possible targets, or part of these pathways can be targets for the treatment of hypoxia. However, it's very important to understand these responses at a molecular level. So we all know from the journal club from last Friday that, for example, the mTOR pathway is quite a complex pathway. So if you're messing around with one part, other things can happen. So you need to understand very well these molecular pathways. So for an example for replacement of oxygen is treat the patient with hyperbaric oxygen. So let patient breathe 100% of oxygen instead of the 20% we're doing at the moment. So you see that the survival is indeed better compared with normal air breathing patients. Again, for the smokers over here, don't do that. This is the proof. Already in 1978 has been shown there that patients who do not smoke do much better compared with patients who smoke. So again, quit smoking. A second example is the example of anaerobic bacteria. So we want to treat patients with bacteria, and we're almost there. Another 2 years and we will treat over here at Master the first patient with bacteria. Which bacteria? Clostridium. And we genetically modify this Clostridium to express an enzyme, NTR, which can convert a prodrug into a toxic drug. So this is the concept of CDET, Clostridial Directed Enzyme Product Therapy. So this is sort of the overview how it works. You have Clostridium, so these are bacteria, anaerobic bacteria, so that means they only survive without oxygen or with very, very low oxygen concentration. So it's perfect. The perfect environment for these bacteria are the hypoxic regions in the tumor. So the growth is only without oxygen, and since they are bacteria, they are sensitive to antibiotics. So you can treat bacterial infections with antibiotics. So this is a sort of rescue measurement. If patients show an adverse effect on the bacterial infusion, you just give them antibiotics 1 week, and it's patient is again cured from the bacteria, not from its cancer. So I said we genetically modify these bacteria to insert an NTR gene, so nitroreductase gene, and this is a gene who can convert a prodrug into a toxic drug. We did a combination of two, made spores of these Clostridium. So spores are not sensitive to heat, are not sensitive to irradiation. So you cannot destroy spores with irradiation, with heat, with cold, so they survive, these spores. So these spores can grow, so they are stable, can grow only in regions of low oxygen. They don't, since they don't grow in normal healthy tissues, there's no immune reaction, and they get washed out from these well-oxygenated tissues. So, when we inject these cells with the NTR gene, these cells, and this is a tumor in a mouse, these cells will colonize the hypoxic areas, necrotic areas, so vessel, viable cells, hypoxic cells, necrotic cells, so no oxygen over here, anoxia. So they will colonize this part of the tumor and grow there. So the moment that we have colonization or tumor colonization in these areas, you can inject a prodrug, which can be converted by this nitroreductase enzyme to an active toxic drug. And having that, you will sort of cure the tumor from inside out. So we did that experiment. So it's not only in theory, it works also. So if you, so you have only to look to the blue and the red curve. So all these are controls. So we start at the time 0 injection IV of Clostridium spores. We wait 5 days to let them colonize in the tumor. Start prodrug treatment. So this is the prodrug, and we monitoring the tumor growth over time. So you see that the combination of these are two different strains. The combination Clostridium with prodrug is sort of giving a significantly higher tumor reduction. So tumors grow much slower compared with the sham and the non-treated tumors. And as last, I want to show you the example of some molecular pathways. So targeting the UPR. So this is again what I showed you in the previous slide. So why is it important? The UPR, mainly the PERK arm of the UPR, influences metastasis formation, influences pH control, also C9, leads to autophagy, and influences also RS redox and biosynthesis of amino acids. So there's a phenotypic consequence of activation of the UPR pathway by hypoxia, and there's a tolerance part of the activation. So what we did is we made in the lab a dominant negative to alpha, so EIF to alpha. So in that sense that UPR gets shut down, or we